How's that? Okay. Okay. Gene, we okay to start now? Okay. Okay, everybody. Well, welcome to the next 2 hours talking about herpes viruses, about a lot sexier topic than we talked about yesterday, but I hope you'll enjoy the information that we're going to give you. I know it's 8 o'clock in the morning, and where's the class president? Where's our class president? Is he here? Oh, he's not here. See that? Oh, I was going to thank him because I knew, as when you get visitors, you always try to make a special effort to, a few more people to be here. And thank you for being here at 8 o'clock in the morning. Thank you very much. Okay. So let's talk a little bit about this group of viruses, the herpes viruses. And we're going to have 2 lectures. The first lecture, I'm going to really focus on the things that the virus has that make it rather unique with respect to the disease that it causes and with respect to the immune response, our ability to respond to infection with this group of viruses. No, the herpes, I said yesterday, the enteroviruses, they're ubiquitous viruses, and most of us have antibodies to the enteroviruses. Well, likewise, for the herpes viruses, they're ubiquitous viruses, and most of us are infected very early on in childhood with these viruses. But now these viruses are different from the ones we talked about yesterday. Number one, it's a DNA virus, double-stranded DNA viruses versus the single-stranded picornaviridae, the viruses that we talked about in yesterday's lecture. Very large viruses, about 4 times the size, as a matter of fact, of the picornaviruses. These viruses have an envelope, which make them rather unique. And in that envelope, they've got these glycoproteins, which has a rather unique role in the ability of the virus to infect and cause disease. But the hallmark of this group of viruses, the hallmark, and the thing I want you to continually remember, is their ability to go underground, to evade detection, to remain silent, to cause latent infections to be reactivated. And it's the reactivation of these viruses which really cause significant problems in medicine and the treatment of some of the patients, many of the patients that you're going to be seeing as you get out into practice. Again, Murray has a great chapter, and I hope you'll have a chance to get to chapter 54 and just take a look at Murray. Very beautiful group of viruses. By the way, is that light? Let's see if we can go down just a little bit. Let's see. Gene, if we hit lights, does that take us? Just take. Okay. Now I'm back on. All lights. Projection? Hit the projection. All right. Is that good? Still light enough for everybody to see? Good, good. Because I think it's worth showing one of the most beautifully appearing viruses that's out there. And that's this one here, the herpes virus. A very beautiful virus, but causing real problems and pain for mankind. In the case of one virus, the herpes simplex virus type 2, almost a quarter of the population are infected with this virus. And we're going to talk about that in some detail. Probably more than 1 million people every year are infected with herpes simplex virus type 2, and a major cause of real problems in sexually transmitted disease. Now, Dr. Margaret Hunt has given you, I know, a nice lecture about the herpes viruses. But again, I'd like to combine the lecture, the structure-functional components of the virus with respect to the diseases that they cause, just to give you a taste in this first hour of why they cause the diseases that we're familiar with. And so, as we did yesterday, we'll break it down into six different groups over the 2 lectures. I'm going to give you a little bit of the background, classification, how do we classify these viruses, the immune response to infection. And here you're going to see that in contrast to yesterday, where it's the cytopathic effect of the enteroviruses, this blowing up of cells, infecting and blowing up of cells, the humoral antibody response is the most important component of the immune response to that group of viruses. Now we're going to change, and we're going to talk about the importance of cell-mediated immunity. Cell-mediated immunity in viruses which don't necessarily blow cells apart, but cause persistent infections, latent infections, and it's the immune response, the immunopathogenesis, that plays a major role in the disease with this group of viruses. We'll talk a little bit about, again, the hallmark, neurovirulence and latency, that I want to really home in on. We'll talk a little bit about treatment and control, but Dr. Richard Hunt, I think later this week, is going to talk about antiviral therapy, and he'll talk about the drugs that are used to treat this group of viruses. And we'll talk, of course, lastly, about the diseases that the herpes viruses cause. Now, at Clemson, all of our students are well versed in the Greek classics. And my Clemson students in here know that, right? Well versed in the Greek classics. And really, herpes comes from the Greek, which means to creep or crawl. And it's a term used to describe the spreading nature of skin lesions. Dewdrop on a rose petal. Dewdrop on a rose petal. We're going to talk about some of these lesions and the characteristics of the dewdrop, which is that pustule that's formed by the herpes viruses, many of the herpes viruses, in particular the herpes simplex viruses. Now, this is an ancient virus. It goes back many, many years. And in one of Shakespeare's plays from the Queen Mab, the midwife of the fairies, describing these the Mab with the blisters, which are the recurrent herpes virus sores that were noticed even during those times. So it's a virus that was an infection and a disease that was described many, many years ago. Of course, they didn't know it was a virus, and they didn't recognize this disease as a recurrent disease as being caused by a virus till the early 1900s. Okay, a little bit of background. There are at least 25 viruses in the family now, Herpetoviridae, okay, Herpetoviridae, and a number of these are known to infect man. Now, in human medicine, what you've got are basically this group of viruses here. And again, those are the ones that we're going to focus on. It's the simplex group, herpes simplex viruses, the Epstein-Barr virus, the cytomegaloviruses, varicella zoster virus, and the human herpes 6 and 7, which cause the exanthum subitum and roseola, 5th disease, the human herpes virus type 8, which is responsible for Kaposi's sarcoma, and we'll talk just a little bit about Kaposi's in the lecture. But we're going to really focus on simplex, Epstein-Barr, cytomegaloviruses, and the varicella zoster, chickenpox, and recurrent zoster or shingles. These are the 4 that we're really going to spend most of our time in. Talk a little bit about this roseola with 6 and 7, and a little bit about 8, but these are the ones that we're going to focus on. Dr. Hunt has told you about this virus, and basically the virus is an envelope virus. Again, it's got those spikes, those glycoprotein spikes that stick out. The proteoglycans on cell surfaces, dextrans sulfate, acts as an initial attachment for the virus. And then there are subsequent attachment sites, the nectins and nectin 1 alpha, which again is an intracellular adhesion molecule, like ICAM1. The nectins are present on a variety of mucosal epithelial cells, but they're also present on neurons. And that nectin 1-alpha is very similar to the receptor that the poliovirus uses in its travels and its ability to infect neurons and those anterior horn cells that we talked about yesterday. So nectin 1-alpha, again, an intracellular adhesion molecule, is also involved in the entry of the virus. And again, I'm not going to go through this, but you get the fusion, and the virus is replicating as a DNA virus in the nucleus of an infected cell. But I do want to quickly tell you about the importance of these peplimers, these glycoproteins that stick out of the virus. They're very important constituents. And these glycoproteins do a number of things. In addition to helping the virus get into the cell, they also allow for fusion of infected cells. And so, in order to evade the extracellular environment, the humoral antibodies, the complement that's present surrounding cells, herpes viruses will cause the formation of cell syncytia, the fusion of cells. And so the virus can be passed from one cell to another, again, without having to get outside of the cell and come in contact with antibodies or other agents that may be present. These glycoproteins also do other things to help the virus. They're made in large excess. So it's not only the glycoprotein that's put on the virus, but hundreds of times more protein is produced than is needed by newly replicating virus, and it's inserted in the cell membrane. Now that's important because these glycoproteins are able to attach to the Fc portion of antibody molecules, immunoglobulins. And when it does that, it creates like a Velcro type of interaction. And so if you've got a virus-infected cell that's surrounded by antibody, what happens to the immune response? What happens to the immune response? What do you think? It's hidden, right? So doesn't the immune response need to identify distinctive antigens on an infected cell to generate an appropriate immune response? Sure. So if you're able to throw a blanket over that so that the host immune response recognizes these cells as being normal, not infected cells, because you've got this antibody surrounding it, isn't that a protection? It is. It is. It's one way, again, that the virus has of isolating itself from the immune response. And again, another interesting way in which these glycoproteins play a role in the escape of immune responses. Now, the virus has a number of other characteristics that are also important. Let me just finish this and I'll get right to you. Some of them are related to these glycoproteins and some are not. But for instance, the virus contains proteins which will shut down interferons. The ability of interferons to shut down virus replication is aborted by some of the proteins that are carried inside of the virus. Okay? The ability to escape this interferon activity, interferon response, is again another important factor that these viruses have. In addition to that, the virus has various proteins which is able to block MHC recognition. Now, Dr. Mayer told you about MHC recognition, right? He's told you about that. And so if you block MHC, what are you blocking? CD8 cell formation, right? And so now you don't get the CD8 response because of the ability of the virus to block the MHC1. Okay? So what I'd like you to remember is that these viruses contain a number of constituents that are very important, not only to its replication, its ability to replicate inside of a cell, but in its ability to abort immune responses to infection. Therefore, allowing the virus to go persistent disease, persistent infection. Now, latency is a little bit different. We're going to talk about the ability of the virus to really go underground, to get into the neurons and be completely protected, hidden from the immune response by its ability to travel down an immunologically privileged area in the body, the CNS, and these neurons that are infected can be latently infected by the virus. Okay, I had a question.
Yes, when you talk about the antibody that's actually the cell, complement, fight the cell, or doing that for the cancer cell?
So the FC-mediated region is actually bound there at that because of the glycoprotein. So the glycoprotein is sticking up the FC, so it blocks that complement binding, so you don't get the lysis of that cell. Good point. Good question. Okay, classification. If we look at the viruses, they're really subdivided into three families. And these three families are based on the genome makeup, okay? So you look at the genomes and you can have distinctly three different classes of genomic structure in the viruses. They're also classified based on the cells that they infect, okay, as well as the cells in which latency will occur, whether or not it's a T lymphocyte, a B lymphocyte, or, in fact, in the case of the varicella zoster virus, a neuron in the trigeminal or the sensory ganglia. And so you have got these 8 viruses here, the simplex 1 and 2, the zoster virus, the virus that causes chickenpox initially, but can be reactivated to cause zoster. All of them are part of this alpha group largely because of the genomic structure of the virus. Now, if we look at the Epstein-Barr virus, it's unique. The genome is quite unique. It's much smaller. But this is the one herpes virus that can immortalize cells. Okay? Makes it a very unique herpes in the herpes virus family. It can immortalize cells, the Epstein-Barr virus. And we're going to talk about some of the diseases that are caused by the immortalization of cells by this virus here. But that makes it very, very different. Human herpes virus type 8 is similar, and it too is a gamma herpes virus, in its ability to cause now Kaposi sarcoma and to cause endothelial cell proliferation because of endothelial growth factor, which is produced following infection with this virus, and the typical lesions that you see in Kaposi sarcoma in individuals who have acquired immune deficiency. The beta virus is really pretty much all to its own, the cytomegalovirus. And this is a virus that is very important whenever you're treating immunocompromised individuals. Those of you who are going to go into transplant surgery, it's a very important virus, and in that instance, one of the leading causes of tissue of graft rejection is reactivation of this virus here, the cytomegalovirus. So we'll talk about that in just a few minutes. Sure. Uh-huh. Right. Good question. Great question. Great question. NK cell activity is believed to play a very important function in resistance to herpes virus infection and to reactivation too. And there's been a lot of controversy, but people have spent a lot of time looking at changes in NK cell activity in individuals and reactivation of the virus. And what some people proposed a number of years ago was that when NK cell activity is decreased, there's a correlation to reactivation of the herpes virus, particularly of simplex, and more reactivation occurs and a more severe reactivation lesion that occurs. So that's a great question, and yes, the NK cell story is a very important one there. Okay. All right, so that's basically what we've got. Now, this next chart really, I think, puts it all together a little bit better. And I'm not going to go through this, but it says, I think it shows you everything that I tried to represent in the previous slide. And again, they're classified based on their primary target cells, whether it's mucoepithelial or whether or not it's a B lymphocyte, a monocyte, or a T cell. And again, that together with the site of latency and whether or not it's a neuron or a B lymphocyte or a T lymphocyte all plays a role in how classification of this group of viruses is done. Now, anagenically, there are differences in these viruses, but there aren't great differences between the herpes simplex viruses. And it's a little bit tricky to find anagenic differences between 1 and 2. If we look at the diagram here, which is in your notes, again, these are ubiquitous viruses for the most part, okay, except for, I want to be careful about saying that with HSV type 2, okay? But ubiquitous viruses, and they do infect various parts of the body. They can cause, in case of HSV1, encephalitis, keratoconjunctivitis, very early on. After birth, very young children will get gingivostomatitis. Almost all of us have been infected with herpes simplex type 1 by the age of 5. Over 90% of us have antibodies. Those of us that don't probably were infected, but for some reason our antibodies have waned. They can cause pharyngitis, esophagitis, tracheobronchitis. They can cause, in wrestlers, for instance, gladiatorium. Anytime you've got an abrasion of the skin, the virus is able to better penetrate and initiate infection. In wrestlers, you see that. Whitlow, what used to be found in dentists and in other healthcare providers, that before the time of using gloves, they would put their fingers in your mouth and they would abrade the ends of their fingers and they would come down with a herpes Whitlow infection. And of course, the one that you're going to have most sensitivity about with respect to your patients are the genital infections that are caused by herpes simplex virus type 2 and type 1. Used to be, we thought that type 2 was responsible for most of the genital infections, but that's all changed now, and that herpes simplex type 1 was only above the waist, but you can see why now. Things are a little different, and you can get type 1 below the waist and type 2 above the waist. Perianal infections is also another site that's often found, and usually it's herpes simplex type 2 that causes that. But down here, the poor infant that is many times at risk of developing very severe infections with type 2, neonatal herpes, and we're going to talk about that. One of the unfortunate things about this group of viruses is, number one, they can escape immune surveillance. Number two, they can cause persistent infections or latent infections. And number three, they can cause subclinical infections. And I want you to remember that one. Because many people, remember I said 1 out of every 4 people are infected with herpes simplex virus type 2. They may never get any recurrent disease, Others, poor unfortunate individuals, will have monthly recurrences of the virus. But there's a third group. And that third group is the group that has subclinical reappearance of the virus. And so where is that most important with respect to the neonates? During childbirth, right? During childbirth. And one of the things, those of you going to OBGYN, you really got to be careful of is, does the mother admit to having some history, or will she admit, or will she tell you about some history of herpes simplex virus type 2 infection. Because simply because there's no lesion there doesn't mean that there may not be virus there. This has raised a big controversy with respect to cesarean sections. And now some physicians are going solely to cesarean sections to avoid any possibility that a pregnant mother might transmit the virus during childbirth. And we'll talk about that. It's an unfortunate situation that is out there. Some of the factors that cause recurrences: UV, fever. That's why you get this term fever blister. If you get fever of unknown origin, sometimes a fever blister can follow. Emotional stress. Those of you taking your microbiology exams coming up here, you know that causes problems. That can cause reactivation of virus. Physical stress, any kind of irritation, emotional irritation. Even foods can cause reactivation of the virus. Immunosuppression, whether it's transient, stress-related, chemotherapy, radiotherapy due to cancer and treatment for cancer, or in those individuals in which they have been infected with HIV and have AIDS. In the immunosuppressed individuals, this virus is a terror. This virus is a terror. And I'm going to show you some pictures of individuals who have recurrences with this virus. Again, cell-mediated immunity is one of the most important components of the immune response in protecting against this group of viruses. I'm sorry, I'm going ahead of myself here with this. Okay, we've covered this. The role of humoral antibody, important, but not as important as cell-mediated immunity. Another group of individuals that you've got to be very careful of are kids who are immunosuppressed perhaps because of a leukemia or because of the therapy that they're receiving for the leukemia. Kids then anywhere from, let's say, 4 to 8 years of age who want to still associate with other kids. That's the age for chickenpox. And if someone with chickenpox gets close to an immunosuppressed child, that can cause tremendous problems and even death in the case of those kids who are treated for a cancer. Their cell-mediated immunity is down. We talked about the NK cells and the role of NK cells. And NK cells and macrophages in particular have been shown to play a very important role in resistance not only to primary infections but to recurrent infections. And again, we've talked about this. The simplex and varicella zoster viruses, they have this ability to lie dormant for a period of time but can become reactivated in the presence of neutralizing antibodies. So once you get the primary infection, even though you've got neutralizing antibody, that doesn't mean that you're not going to be reinfected or have a recurrent disease. And most studies indicate that the sensory ganglia are the source of virus that produce recurrent skin lesions, and in particular the trigeminal ganglia in type 1 and the sacral ganglia in type 2. Here's the gift that keeps on giving. So once you're infected, you've got it for the rest of your life. No antiviral that's known right now is going to cure you. You'll have this virus for the rest of your life. Whether or not it's herpes simplex or the Epstein-Barr virus or cytomegalovirus, this virus stays with you for the rest of your life. And the key is to learn how to control these recurrent infections, and they can cause severe problems. The hallmark of many of the viruses is this ability to cause latency. Now, latency, how does it happen? There's a primary infection, mucosal epithelial surfaces. Now, I showed you that beautiful virus with that wonderful envelope with those glycoproteins that stick out of the envelope. If the integrity of that envelope is broken in any way, that virus is dead. Okay, it has no chance of causing an infection. To be infected with the herpes viruses, most of the herpes viruses except for the varicella, and the herpes simplex viruses in particular, you have to have close mucosal contact. Okay? Mixing of mucosal surfaces is the primary way in which many of these viruses gain entry. Okay? You can't get herpes from sitting in a hot tub. You can't get herpes from sitting on a toilet seat, although there's been many law cases that said that I got herpes because I was in a hot tub with so-and-so and they had the herpes virus. Basically, it's not possible. I won't say it's completely impossible. Basically, it's not possible. Why? Because that envelope is very fragile, very sensitive, not only to the water, to the heat, but to the environment, to UV, to drying. As soon as that envelope breaks apart, that virus is not infectious. You have to have close mucosal contact. You've got to work at it to be infected with this virus. Okay? The primary infection is a lesion. Dewdrop on a rose petal. And again, I'll show you some of those lesions. That's the primary infection. You get these blisters. Okay? In the case of herpes simplex genital infections, you get blisters around the genitalia, perianal region, inner thighs. You get these blisters, a real soreness. It will subside in about a week. The blisters will crust up. You're still infectious. You're still infectious. But then when the crusting ends, you get re-epithelialization of those mucosal surfaces, and you're no longer, theoretically, no longer infectious. But that's not the end of the story. It's not the end of the story. Because while the host immune response may result in production of some humoral antibodies, this virus now goes from those mucosal epithelial cells underground and gets involved in underground or guerrilla warfare. The ability to get to those immunologically privileged sites, those peripheral neurons, get into those cells and hides out. Okay? Hides out. And it just stays there. And it stays there because it forms, we think, an episomal type, creates an episomal form here inside of the nucleus of a neuron. And remember, we talked about that nectin-1 alpha as being one of the receptors to allow it to get into neurons. But once it gets inside this episomal form, and the only thing that it does inside of the neuron, for the most part, is to code for a small number of replicative enzymes so that the episome continues to survive, okay, inside of these neurons. The full genetic sequence is here, but not all of the genes are being transcribed. And only a small number of them are being transcribed, and these are the latently activated transcripts. Okay, latently activated transcripts. Just a small number, enough to replicate this. Until something happens. Maybe a month, maybe a year, maybe 5 years after your initial infection, you have a very stressful situation in your life. Maybe it's not so stressful. Maybe it's only menstruation. Maybe it's laying out on the beach and having UV lights somehow caused something to happen. Now, what is that something? Still a black box. Still a black box. Some people say that there's a neuron growth factor that's activated, and when you get neuron growth factor, nerve growth factor activated, that it causes this episome to be fully transcribed, and now infectious virus to be produced inside of this neuron. And when the infectious virus is produced, it's then going to travel down those peripheral nerves to the initial site of infection, those initial mucosal cells that were infected during the primary infection and cause reactivated disease. Another course of the same primary infection. Usually it's a milder infection, probably because you've got some immunity to control the infection at those skin sites. Again, this is the hallmark of this group of viruses. Now, there's two hypotheses. One is that, as I've just shown you, the virus is in a static state. That is, it infects these neurons, and it just stays there. It goes to sleep until it's awakened. Some people say, that can't be. Why? Because we know that somebody was out on the beach yesterday, and today they're starting to feel a tingling sensation, which to them means they're going to get a recurrent cold sore. The time that it would take for the virus to go from the sensory ganglia, the trigeminal ganglia, to produce infectious virus, to go down the peripheral nerves, to cause a new lesion, is thought to be more than just 24 hours. And so is it really that UV? Is it that is causing the virus to be activated inside of a cell? Or is there another hypothesis? Is it possible that the virus continually replicates inside of those neurons, continually making new virus, and continually being shed to those mucosal surfaces, so that there's virus always there, but your immune response is able to control the virus when it gets to those skin sites and no disease occurs? But when there's UV, does the UV somehow affect the immune response? So prostaglandins are produced, they shut down natural killer cell activity. And so now at that local site, is the virus able to take hold and now replicate and cause a new lesion? Okay? Two thoughts, no one has proved either one. You could have continual shedding, or you can have a true reactivation in which the virus perhaps very quickly replicates inside of these neurons and then falls down this peripheral nerve to cause the new lesion. No one's sure which one actually occurs. And this just diagrammatically shows that, what might be happening in the dynamic versus the static. So in the dynamic, there's this continual trickling of virus, low levels of virus, but it's controlled at the skin site. But maybe UV, maybe menstruation, hormonal changes, change the local immune response at the skin site, and so now a lesion can occur. In this case, the static state says that the virus has to be reactivated inside of the nucleus. There isn't this trickling effect, and the peripheral nerves don't receive any virus coming down here until whatever signal comes about inside of these neurons to send virus, new virus, down, and then reactivation causes an acute infection at that initial skin site. Again, no one knows what's happening there, but it's important to know that the virus is there in those sensory ganglia. It hides out. Somehow it comes back. We've talked about this latent activated transcript and latency-associated transcript. And it is really on both the unique ends of the unique long and the unique short ends of the herpes viral genome. There is this section here, which is the latency-associated transcript, which maintains the virus inside of those neurons. That's a very important feature because perhaps, if you're talking about antivirals, perhaps you want to be talking about antivirals that could target this latency-associated transcript. Is there a way that you could prevent this from being made? And perhaps you could come up with a new antiviral. That's going to be a difficult one, a real difficult one to do, but people are looking at that. Question?
I don't know if you want to talk about this later, but do they ever cause damage to the nerve?
Oh, yeah, good question. Good question. So we're going to talk about post-herpetic neuropathy. Post-herpetic neuropathy. And in many cases with shingles, okay? When the dermatomes, complete dermatomes, have this reactivation of virus, those neurons can stay agitated, aggravated for months, for years after those initial lesions. And so, yes, there can be neuronal damage there. Yeah. 'Cause, 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 Yeah, that's a great point. And actually, I'll go back to the woman who is about ready to give birth to a child. There's a lot of work that's been done to look at secretions, and using PCR technology, you can find in people who have had previous herpes virus type 2 infections, you can find virus in secretions. You can find rather large amounts of virus in the secretions, which goes back to your point, this continuous shedding of virus. The second point to support that, where most of us are infected by herpes simplex virus type 1 very early on, you know, probably gives it to us? Mom and pop, grandmom and grandpop. That's saliva and the transmission of virus in saliva. Even though we don't have any cold sores, the virus can be shown, again using sensitive PCR techniques, to be present in most of our saliva, HSV type 1. Again pointing to the fact that this continuous shedding may actually be occurring. Personally, I think there is something to that. The real question is, what is it, though, at the local site which allows for the virus to cause disease, to cause the symptoms of the disease? Okay? Did that answer your question? Good. Okay. We've talked about this. And what I would like to... Boy, Dr. Mayer is really going to be proud of me. You know that? He is really going to be proud of me because I am going to finish this part, the first half, right on time. Even though that's a little bit fast, Dr. Mayer. That clock is a little bit fast.